It is Dr. Sheila Tikku speaking about nitrogen fixation for class 3rd year. Nitrogen fixation of class 3rd year is in bioinorganic chemistry, important part of bioinorganic chemistry. Element nitrogen is relatively unreactive. In order to fix nitrogen, that makes nitrogen react with a substance to produce nitrogen compounds. It is generally necessary to use energy rich conditions. High temperatures are critical and necessary to produce nitrogen. These are usually found in the buds of beans. Beans means rhizobium enzyme which fixes nitrogen. Bacterial nitrogenase system. In the bacterial nitrogenase system, we have important N15. This N15 is fixed with molybdenum, essential for bacterial nitrogen fixation. Since this function can be turned off and only removing and restoring molybdenum in the environment. Magnesium and iron are also essential parts. In 1960, the first active cell free extract was prepared, and since nitrogenase, the enzyme are called obtained in condition of bacteria. In the case of bacteria, they were produced and they were taken into use. This was happened in long ago when nitrogen was fixed with mercury and we had ammonia in the presence of hydrogen gas at the high temperatures. Synthetic nitrogen fixation systems. The discovery of dinitrogen was unable of forming table complex, and this table complex was formed by molybdenum, and this produced products of nitrogen. Then we had diphenyl phosphine ethane. This diphenyl phosphine ethane produced at higher temperature acting reducing agent as Grignard reagent. This Grignard reagent was in use in vitro nitrogen fixation. This in vitro nitrogen fixation in anaerobic pasteurmonia and Clacidonium. Further, in Calasia and such type of compounds which have nitrogen fixation with rhizobium species. The active enzyme nitrogen fixation in nitrogenase, it is not a unique species. Some unique species or the species of beans are very popular and very conditional to fix nitrogen. It seems that the active site for nitrogenase binding involves the molybdenum atom. It has established EXAFS. The ultimate source of adequate capacity is pyruvate, and the electrons are transferred from via ferredoxine to nitrogenase. The whole cycle is given on the next page with a table. A systematic diagram for the production of fixed nitrogen compounds, including the source of material and energy, and the overall reaction are given in the table 7.5. Note the presence of hemoglobin. This is monomeric oxygen binding molecule which helps in the binding of ATP, and it also helps in the binding of blood cells in human parts. The lithium and any oxygen that is present is very tightly taken as the source of nitrogenase, which can be a cooperate in the presence of oxygen. On other hand, it allows a recovery of oxygen for respiration of different type of compounds.